Hi there. Sorry for the delay there, but we had some webcam problems, but I think I get it all going right now, and hopefully you can see me online and that you could hear me. If you can't hear me online, please send a message through texting on the YouTube channel, and we'll try to fix that problem. So welcome to the joint presentation of the Botany Seminar Series and the Plant Conservation Alliance. To those of you here and watching online, the Botany Seminar Series is a monthly and soon to be bi-weekly series of presentations hosted by the Department of Botany at Smithsonian's National Museum of Natural History. The Plant Conservation Alliance, or PCA, is a public-private partnership among 12 federal agency members and over 380 non-federal cooperators that share the same goal to protect native plants by ensuring that native plant populations and their communities are maintained, enhanced, and restored. PCA holds bi-monthly meetings in and around Washington, D.C. that are an open forum for anyone interested or working in plant conservation. After a featured speaker, PCA holds a roundtable for attendees to share relevant events, as well as updates from each of the PCA working groups and committees. So for those of you in the room today, everyone is welcome to join us after today's talk from 3 o'clock to 4 o'clock here in this room for the PCA meeting. For those of you online, you can call into the phone number 1-877-414-1336 using the passcode 6177335#. During today's talk, if you have questions, please comment on the YouTube site, and I will read your questions to our speaker after his talk. So now, after all that introduction, it gives me great pleasure to introduce Chris Martine. Chris is the David Burpee Chair in Plant Genetics and Research and the Director of the Manning Herbarium. He received his BS and MS from Rutgers University and a PhD in Botany from the University of Connecticut. Chris studies the plant family Solanaceae, including the unusual breeding systems of the genus Solanum in northern Australia. He also explores questions related to seed dispersal, invasive and native species interactions, and pollination biology. He has a very strong interest in producing outreach materials that are effective in generating public interest in science and in the organisms we study. He often takes to Twitter, YouTube, and other online mediums to spread the word about natural history and how cool nature can be. And you'll hear a lot about that today. Just as a side note, in 2016, Chris received the Passion in Science Award from the New England Biolabs. And just last year, Chris received the Peter Raven Award, which is given by the American Society of Plant Taxonomists to a plant systematist who has made an exceptional effort at outreach to non-scientists. So today, Chris will be talking about plants are cool too, #SciComm, media relations, and a botanist on Mars. Chris, thank you.
Thank you so much. Thank you, Gary. I actually forgot that you were talking about me there for a minute. And like I just zoned out, and I was like, wow, that guy sounds like somebody I should talk to. Like, we have a lot of common interests, and then I remembered you were talking about me. That's where I'm at about this point after being here for a couple of days. So thank you for having me. Hello to everybody out there. I'm counting on the folks that are actually in the room to laugh at all the jokes. There are about 200 people here, so it's going to be loud when they do laugh. But you can also laugh remotely. We might hear it here in DC as well. So yeah, we are going to talk about this stuff here, right? And so we do a lot of things in my lab. You can see a lot of the things that we do here. When we get lucky with all those things we do related to evolution and plant reproduction and ecology and natural history, when we get really lucky, we end up discovering and describing new species as well, particularly in Australia in the genus Solanum. And we get a real kick out of telling people about them. And that's the sort of thing that I'm going to really focus on today. There's a lot of things there's threads that sort of run through all of this work, but two of the primary things are that we are a botany lab. We study plants. The students that I work with are proudly botanists, and most of those students, is the second thread, are undergraduate students. So at Bucknell University, where I teach, we're a primarily undergraduate institution, and so most of the students that come to the lab are also that way, even though we do have a small master's program and some outstanding students in that program. Another really common thing that runs through all of this that I need to point out, as I did when I gave the Philo Pizza talk last night, is that all of the support that have that helps us do all these things comes from an endowment at Bucknell, money that was donated by this fella, David Burpee, upon his passing in the early 1980s. He's the grandson of the founder of the Burpee Seed Company. We do not have an actual formal relationship with the Burpee Seed Company, but the name is still there. And as I said last night, I'm only about 5 years away from walking around campus holding a marigold and smoking a cigarette. So that's the kind of thing you do as a senior faculty member or the grandson of a seed magnate. All right, so there's that. So I'm very grateful to have this position. It's a really unusual thing to exist. When I first heard about it, I didn't even believe it was real, but it's an endowed botany professorship at a small private liberal arts school. So I feel so fortunate to have that job. Super lucky. I'm going to use the words we and our quite a bit. And when I do that, I mean students primarily. You can see most of those students that have come through the lab the last couple of years and done really good work in the lab are undergrads, and a couple of master's students, Cheyenne and Jackie. And I also have to give immense amounts of credit to the current Burpee postdoctoral associate. That's Dr. Angela McDonald, who's been with me for a couple of years and is actually moving on to the Chicago Botanic Garden next fall. Outstanding scientist, and I'm so lucky to have her in the lab as a scientific partner. So when you look at that list of students and you see all those students that are really interested in botany, at least for this window of time that they're in the lab, you might say, well, that's cool. Botany must be doing really well, but you're also a lot of smart people in here, and I recognize a lot of faces there, people that also know a lot about the field of botany. If you think that there's lots of students that are really interested in plants, then apparently you might be wrong. This is an article from just a few years ago talking about, well, you can see the headline, Death Knell Sounds for Botany Degrees. This is a story about a declining interest among young people in studying plants, and thus then a declining demand for plant courses, and maybe a reason why administrations might then not run those courses and maybe not have those faculty lines and maybe not hire PhD botanists to teach those courses, right? And so this is kind of a bad thing generally, right? In a world where we got to do lots of things, including feed many people, a decline in the knowledge we have about plants is not the best scenario we could consider. So they do a number of things in this article where they say, here's some ways we might want to solve this issue, right? This issue of declining interest in plants. And one of the things they say that we should try doing is we should teach botany by stealth. And what they go on to say is that what we ought to do is, even when students aren't interested in plants, we should sneak it into the curriculum. Just when they think they're not learning any botany, you fake them out and you teach it to them anyway, right? Now, lots of us have heard this approach, right? And probably tried it, right? And I've been told many, many times that, what you do is just tell pre-meds that medicines come from plants, right? And then bam, they're hooked. They're botanists for life at that moment, right? This is sort of the general concept, right? And I would say that it's something we actually have been trying to do for a long time. And it kind of doesn't work. Right? It kind of doesn't do the trick. Now, I don't want you to trust me on that. I want you to trust the data. I did a very careful data collection exercise. And what I mean by that is that I looked at Twitter, and thank you. And I use Twitter quite a bit, like some other folks do. We were told not to say anything. So here's what I did. I looked at Twitter, and I sort of said, what are students saying about their botany course on social media? Right now, of course, there's folks in here that would say, well, Twitter's not the best place to do that. There's one generation that would say Instagram's the place, and my son, who's 14, doesn't even have an Instagram account, right? They say Snapchat's the only place you're going to look for what people are saying. But I looked at Twitter for a few years and saw, what are students saying about their botany classes? Let me show you some of this important data that I collected. All right, so here's one here. I seem to love plants, but I changed my mind when botany came along. I was really into this subject, and then I took a class. And now I don't like it anymore. How about this one? Can I please skip botany today? Go back to bed, please. So students could say this about a lot of courses, but in this case, students say botany course. How about here? Shoot me. Hashtag botany. Hashtag boring. Hashtag no friends. Yeah, I don't know whether that's there are no friends in the class, or once you take the class, you no longer have any friends. Let's try this one. We learned so much in botany class. Hashtag said no one ever. How's that even possible? That does hurt, doesn't it, right? How's it even possible that you could take a botany class and feel as though there's nothing to learn? Or this one, botany is so irrelevant. You're in a class about plants, and somehow you are led to believe that the subject matter itself is irrelevant. Yeah, that hurts a little bit, right? Who knew plants could be so freaking confusing, gang, right? If you're adding consonants, G's in particular, to the word confusing, you know you're pretty frustrated, right? So I would say that it's not working. You know, this whatever approach that we are using in lots of our classrooms, and not the case for everybody, lots of us are also pretty decent instructors, right? But something about the way we've been teaching plants is not really working at present, right? And maybe some of that is this attempt to sort of go with this stealth approach. So, you know, if that's not working, what's going to work, right? And what I think we need to think about is more ways to put plants in front of young people's eyes in positive and dynamic ways, right? And one of the ways that I've been trying to do that is through this web series that Gary mentioned briefly. Plants Are Cool Too is a YouTube series that's, you know, really helped, Botanical Society of America really helped get it off the ground. It's been sort of sponsored by other partners along the way. But this is a series that was meant to sort of focus on cool plants and the people who study them. So sort of this dual idea of saying that plants are really amazing organisms that you want to learn about. And you know what? There's also regular people that are interesting and dynamic that study them and you want to know about them too. So I'm not going to go through the whole series, but I will say that we've made something around 10 episodes and a couple of other short episodes. And some of them include, of course, a carnivorous plant episode shot in Louisiana with Maggie Haynes, who's now at Eastern Michigan State. We did this awesome episode at the Clarkia Fossil Beds in Idaho, where they're pulling out actual leaf tissue that's 15 million years old, not just impression fossils, but actual tissue. We spent some time out at White Sands National Monument, filming a couple of episodes, including this one with my friend Chris Martine, who's at Chicago Botanic Garden and hopefully watching live right now. That was a ton of fun, looking at hawk moth pollinators on that same trip. Also filmed this episode with Mike Moore and colleagues, looking at gypsum endemism, also really fun, an opportunity to talk about how speciation happens. Okay, we'll use that a lot in my courses to show how that could happen. And then a couple of the more recent ones were filmed actually in Pennsylvania, where I teach and live right now. One of those was this one, shot in Cole Township, Pennsylvania. This is the only episode we have where we don't feature a scientist. All the people in this episode are volunteers, are conservation volunteers. And this one was really therapeutic for me to make, because this was a video about coal land restoration, lands that were devastated by strip mining that are being restored by folks who want a place to do recreational riding, right? And what they are doing is rebuilding a forest on absolutely devastated habitat. So for me, this was really therapeutic because it's a couple of groups that you wouldn't seem to think would have commonalities, right? So it's people that are recreational riders, like ATV riders, and people like me that they might call tree huggers, coming together and saying we actually have some common values, some places where we can work together, do some positive things. So if you need a story that helps you feel better about that sort of stuff, this is a good video to watch. And then the most recent one, which is one I'm going to come back to at the end of the talk, was shot at a place called Shikellamy Bluffs. You see those cliffs there over the Susquehanna River. And we'll come back to that one in the end. We're also, just at the right time, Warren, you walked in, we're also heading to Hawaii next week. And we are going to work with Warren and a bunch of colleagues in Hawaii to shoot some one video on Shedia in Hawaii and look at sort of island speciation kind of ideas, island diversity. And also spend some time at the National Tropical Botanic Garden. So that's sort of what's coming next. And you know, there's a lot of reasons why I make these videos with my colleagues Paul Frederick and Tim Kramer, who are the professional video people who I hired to do this work with me. But one of them is that, you know, these are organisms, like lots of organisms that aren't sharks and panda bears. These are organisms that really could use some help having their stories told. My friend Uzai Sezin, who runs this awesome website called naturedocumentaries.org, sort of like this warehouse of nature films, really said it well, I think, when he said the natural history of plants requires powerful storytelling. And that's sort of the idea that we are latching onto for the video series, but it's also the idea that I really latched onto for the things I want to spend the rest of my time talking about, right? And what one of the things that has inspired me to sort of tell this particular story about us sort of working with the media to tell our science stories actually is pretty well embodied in this Twitter poll here, right? So here's the story. A journalist interviews a scientist about their research. The journalist then goes and writes the story about the research. And the poll question is it required that the journalist then come back to the scientist and say, can you check the story before I publish it, right? And then you see what people had to say, right? And it turns out, you know, when it wasn't hard to do the math, right? I figured out that 82% of the scientists who responded to this poll, and the poll had over 4,600 people participate, participate, said, yes, if you interview me about my work, I expect to see the story before it goes to press. And that is not how it works. Right? Not how it works, right? If politicians were doing this, we would not be satisfied, right? And yet, many scientists really wish this is how it would happen. And there's lots of reasons for that, right? Lots of scientists have been burned before, right? That their work wasn't presented in a way that we might have felt was rigorous or accurate. Our quotes may have been may come out in a way that we don't feel is rigorous or accurate. So we kind of want to look at it before it goes to press because we want to make sure our work is well represented. I totally get it. And that's also a fundamental misunderstanding of how journalism works, right? So how do we deal with this, right? So one of the things that I've been talking to people about is that now more than ever, we actually have an opportunity to control the stories that get told about our work, control the narrative in a way that we have never had in the history of humankind, right? We have this ability to get our work out in our own words in a way that we haven't had for centuries, right? So what I want to tell you about are some ways that we've been trying to do this with some of the outcomes in our research lab. I want to be careful to say that the starting point for all of this really is still doing good science, right? Producing actual good science work. But then there's some work we can do as individuals where we actually choose some of the stories that come out of our research. We do some of the writing about it and the sharing about it in our own words, and we sort of provide that information to people in the media and people that are sort of putting stuff out there in the world. And it is going to require that we actually be willing to do a little bit of additional legwork if we want our stories to be told in the ways that we most appreciate them, right? So let me tell you about two things that we have done, very intentional processes that we have engaged in to sort of test out some ideas about what would happen if we begin to tell our own stories, if we actually provide narratives to media about some of our scientific outcomes. So two case studies I want to share, and both of them have to do with what Gary mentioned, which is this group of nightshades in the genus Solanum that occur in Australia. Beautiful plants. I did this yesterday, I'm going to do it again. I just want you to stare at that for a moment. Gorgeous. Get lost in the corolla. That's very nice, isn't it? Yeah. All right, so that's one of the reasons I study this group is that they are very attractive, but they also have a lot of neat stuff going on with the flowers, and I told a lot of those stories yesterday. The group that we study is a group called the Solanum dioicum group. It lives across the northern part of Australia. There's something like 28 described species in the in-group that we study. And as I mentioned earlier, that number is increasing in part because we know this group so well that every time we go back, we find something different. I'm hoping to be on my 7th trip in the last 15 years to that region of the world this summer. And I've got my eye on some things that I think are already species that need to be described based on the herbarium material. And so the germination, you see what I did there, of this idea that we could sort of take advantage of this ability to communicate our own work to the world through the media really started with this species that we described some time ago. The species we eventually named Solanum cowii after the curator of the Northern Territory Herbarium in Darwin. And I was there with my colleague Beth Capaldi and our student Gemma, and we found a number of specimens that led us to name a new species. And one of the more interesting parts of this story was that I was only able to give a name to this thing and actually get the material required to name this because I found a bunch of great specimens in the herbarium that had been annotated by a fellow named David Simon, who's sort of the leading solonologist in Australian history. And he had left such a detailed set of notes in the folder along with this undescribed taxon that I made him a co-author on the paper, even though he had been dead for over a decade. And that sort of notion there actually caught some attention with the science media, and the story sort of blew up more than we imagined it would because of that, and got us a lot of buzz about that story. And it made me think, this is the kind of stuff that people respond to, right? New species discoveries in particular, especially when there's something interesting, there's sort of this human element, something that's fun about the story, might be opportunities for us to generate excitement about biodiversity science in general. So I thought the next time we have an opportunity like this, I want to try a few things. Right? So the first thing that we tried it with was this species that we eventually named Solanum watneyi. The specimens that we collected that allowed us to name this species were collected in 2014 with my family, my daughter Izzy, my wife Rachel, and then Jackson, who you see here, who's now in 8th grade. That was a picture, I said this picture was taken only 2 weeks ago. He is now 6 feet tall. That's how it goes. You like that joke? Yeah. 5'11". Okay, fine. Yeah. All right. So this was a cool thing. You know, that wasn't the story we told, but it was nice that we collected it while we were all together on a 6-week expedition. So we knew we had this species that we needed to give a name, and we had talked about how could we make the next species something that might generate some media buzz. And right around that time, the movie The Martian was coming out, based on the book The Martian. And I said, wow, what a cool opportunity, right? When is there ever going to be another time where we have a blockbuster film that features a space botanist played by Matt Damon? And I realized, okay, that's never going to happen again, right? So maybe we should take advantage of this. So I came into the lab, I said, what do you think about this? And we all said, okay, let's do it. Let's name this species after that character, Mark Watney. So that's what we did. We thought, let's see what happens if we sort of carry this. Maybe we all push it a little bit on social media, see what kind of things come out of that. Can we get a buzz about this, maybe even eventually drive some traffic to the actual paper? The only problem was that we actually hadn't written the paper yet. So we made this decision, the movie was coming out that weekend, and we had no actual scientific thing to share. So at the time I was doing a lot of blogging for the Huffington Post, so I decided I'll at least just try this. So I wrote this piece, why name it a new plant species after the Martian, got the sort of thing out there in time for the opening of the film, and then we took a plant of Solanum watnii to the movie on opening weekend. There we are as a lab group. If you're wondering if you can do this, there's your answer, right? But we went up with a potted plant to hand in our ticket, and the person said, I don't think you can bring a plant into the movie theater. And I said, has anyone ever tried? And he said, no. And I said, well then, right? So we did bring the plant to the movie, and as you might imagine, it loved the film. Very much into the film The Martian. Right, so we did that, which was cool. That got us in the local newspaper. There we are, right there, Hollywood twist given to Discovery. You know it's big in our hometown when you share the front page of the local section with kids in sumo wrestling costumes. Big time there, right? And that somehow ended up getting us a mention in a BBC News article. So, here we are in a little small bit here. The headline is that somebody was offering the naming of a moth on eBay, and that got the headline. And then they said, if you think that's interesting, well, there's also this new species being named for Mark Watney. It was the opening weekend of the film, and we thought, boom, like we did it, everybody, way to go. Good sort of thought experiment. Like if we put stuff out there, if we catch the sort of pop culture thing, maybe we can get attention for our discoveries and for biodiversity discoveries in general, and we were very happy. Right? And then something happened right around the time that the paper was actually going to come out, and that is that The Martian was not only popular with audiences, but popular with critics, and it got a Best Picture nomination. So here we were with the paper about to come out in the open access journal PhytoKeys, online only, and here we were approaching the weekend of the Oscar ceremony. So we thought, well, maybe we can actually catch the buzz of the actual Oscar ceremony. How do we do this? So we worked with the communications people at the journal and the communications people at my institution at Bucknell. We wrote a press release together where I actually supplied all of the actual scientific information in my own words in this press release. And we sent that out on the Friday before the Oscar ceremony. The paper came out online the Friday before the Oscar ceremony, and we thought, okay, now what, right? So we got some of the basically good science stuff, like Eureka alerts and things like that, where they're sort of taking the same press release you put out and regurgitating that. Great, that's fine, right? And then local news outlets all over the country sort of start to catch note and put it out there. Places like the Durango Herald taking our press release and just reprinting it again with my words about the science surrounding this story. Other local newspapers around the country, like the Daily News. My father's from the Bronx, New York, so for him, the Daily News is about as good as it gets. So we were all very excited about that, right? Local TV news affiliates pick it up all around the country, and then it goes international. So here we are with this story. I'm not sure what a vet and shopper is, but I'm excited to be one. Probably, I think it means botanist. I think, yeah. And and then it comes back to domestic markets. And instead of the press release just being reused over and over again, we start to get original articles written about this species, Solanum watnii. Time Magazine picks it up. And one of the lovely things is that we also provided a bunch of images with our press release, images that we supplied that we took. And one of those was of Emma Frawley, who was an undergrad at the time, who's just going to start her PhD at UW St. Louis next year. And she was a co-author on the paper. So there's Emma working with the plant. And on the day that the story breaks, her mom is visiting campus. We're sitting at a local cafe, just hitting refresh, and we're seeing her image pop up in news outlets all around the world. It's actually very, very exciting, right? Lots of other original news articles. Live Science picks it up. Discover magazine writes this article: 5 things Matt Damon has in common with the new Mark Watney plant. Number 3 was that they're both very cute. Tech Times writes a nice story. GeekWire picks it up and then is the only outlet that says, we've seen this before. So this is the second time that they've heard about it, but they still write us a nice article about this species. So we were very excited about it, right? It was thousands of outlets pick it up. If you had searched Solanum watneyi around that time, you would have found over 2,200 different news outlets were running a story about our new species from Australia. And for me as a scientist, and for lots of us, right, we want to know also is this mean people are looking at the actual science, and they were, right? So around that time, if you had gone and looked at the actual article views on our journal article, we're over 4,700. I know that the smart of you in the room know that total views is different than unique views. So that's actually 2,800 unique views and just shy of 2,000 me views. Going back to the paper, reading it over and over again. I'm just kidding. I couldn't do it 2,000, but it's 500. So yes, the traffic was going back to the original paper, the original source material, which I was very excited about. I was also very excited about this outcome because now that we are mentioned in the Wikipedia article about the Martian, we know that we'll be cited by every 5th grade kid who ever writes a book report on the Martian. So that was pretty cool. But for me, my most fun outcome of this was this right here, right? And so all that news coverage, all these other things that came out of it, and this is the one that warmed my heart the most, that somebody who was obviously a really big Matt Damon fan found out about Solanum watneyi and made a painting, right? And then posted it on Twitter and tagged me and said, I made this. This is one of those interactions that as a scientist, maybe perhaps 20 years ago, I wouldn't have had, right? But now we have this sort of global ability to talk to people in all sorts of different ways and have this sort of thing happen. So I was really psyched about this particular interaction. So we were really happy with how this all turned out, right? And we thought that's a successful media relations story. Good job, high fives all around, right? And a short time after that, we had another species that we needed to work on. And we had sort of felt like we did the pop culture thing. We all know that, like, if you tie a new species name to some pop culture hero or phenomenon, you'll often get kind of a little bit of buzz for it. We've seen really good examples of that in lots of people's labs. So we decided to try something different. So we had this species, and we were thinking, what are we going to call this thing? And right around that time, I got an invitation to come and give an assembly at the local middle school by a teacher named Brad Catherman. And Mr. Catherman said, can you come in and talk to my life science students about biodiversity science? We're covering this kind of thing right now. And I said, sure, I'll come in. I have some slides I can show. I got all sorts of things we can do. And he said, well, I would really prefer if we did something more hands-on. And they told me he had 150 students. And I thought, what can we do that's hands-on? And then bing, light bulb goes off, and I think, we're looking for a name for this species. So what we did is we challenged those 150 students to each write an essay coming up with a name for our new species. This is after I went in and gave a talk, showed them real plants, gave them all the natural history notes they would want, gave them a little bit of a primer on botanical Latin, and then they sort of went for it. And in the end, the name that they chose was Solanum ossicruentum. Has a lot to do with the fruits. So when the fruits are unripe and you cut them open, they kind of look like they're bleeding. And when they're fully mature, they're as hard as a bone. And they really like this sort of juxtaposition of sort of bloody and bony. And that's where the ossi for bone and the cruentum for bloody came from. And here's most of the students that helped us come up with that name. And at that time, I thought, okay, this is cool, right? This will be a thing that really pushes this story forward. People are going to really be into this. We actually waited on the paper and the press release to come out on National Teacher Appreciation Day, and I thought this is going to be a really good story for us, and we sort of ran with it. And early on, places like Science Daily picked it up, and in large part focused on the student component, right? What a cool thing about these students contributing to this. And there's, in the past few years, also been some others that were named by kids, by students that got a little bit of press as well. And the early stuff where they're taking the press release and regurgitating it, like on phys.org, is doing that same thing, focusing on the students. But by the time it goes through sort of local domestic markets and goes international, the focus turns back to what the students actually were most interested in the first place. And so by the time it shows up in the Iran Daily, it's really focusing on the fruits, right? The fact that the fruits look bloody and that they harden to be very bony, right? Which was kind of a neat thing, right? The biology sort of ended up trumping some of the other things. So it comes back to domestic markets. We get original stories like this one about sort of recent new discoveries, and they're talking about sort of these spiky and bloody tomatoes. We end up on a number of websites that are resources for students to learn about natural history, like Love Nature and Student Science. And now the story is that it's a terrifying tomato that appears to bleed. It's covered in these crazy spines, right? And this becomes the sort of leading story for this unusual plant, right? Science News, scary tomato appears to bleed. This resource for people learning English as a second language. So it's current events that people can read and learn English from, right? And scary tomato that appears to bleed somehow makes that list of current events, right? Bedtime stories for curious kids picks it up and has this story for a kid to read about before they go to bed at night. But the real turning point for us was when Atlas Obscura picks it up, right? So this is sort of a weird news site. Lots of you have seen stuff here. There's often weird science news here too. And they really go all in on the terrifying bleeding tomato. And that's what gets the attention of NPR Science Friday. And we get assigned this really incredible science writer named Chow Tu on the West Coast who writes this really nice story. That gets us on PRI, that gets us in all sorts of additional local NPR affiliated stations. And that's awesome, right? So we were pretty psyched. But what that really got us was the place we've been wanting to get to this entire time, vampires.com. Vampires.com writes the story of the vampire tomato. And this is really, to me, indicative of the sort of thing that places where your science can end up if you're just willing to sort of get it out there a little bit, right? Eventually, you end up in these places where nobody would ever hear about your work, right? So people who really like vampires now know about a new Solanum species that was discovered in remote parts of northern Australia. Right? Then maybe they never knew that was possible to find that sort of thing before. Right? Again, though, for me, the most heartwarming thing was people making art and posting about it. So here's an illustration of Solanum ossicurentum posted on Instagram by this person saying, "Hey, I heard about this plant and I thought I'd make a sketch of it." Right? Their friends will see it and they'll all maybe be inspired to think more about plants, more about biodiversity. So did it do the same thing as the other work, right? Did it get people to read the paper? Yep, around that time, 1,700 views on the paper. If you had searched the Latin name, you would have found over 1,700 news outlets covering stories about our new plant. And then something really awesome happened to this one also, which is that ESF put it on their top 10 new species for 2017, one of the 2 plants they put on the list that year. That generated all sorts of new buzz. It got us on ABC News, sort of the NPR equivalent in Australia, great story in the Philadelphia Inquirer and lots of other places. And that really increased the impact of the paper. So as of just a few weeks ago, we're approaching 9,000 views on a paper that, you know, legitimately maybe 10 people would have read otherwise, right? Like honestly, who's reading my new species papers? Generally, not a lot of people, right? And so here's a way that we're really spreading the word about the biodiversity discovery. Just a few weeks ago, you would have found about 6,600 places online where they're still using the name Solanum ossicurentum. Just outrageous. I just can't get over it, honestly. It was successful in a number of ways, so much so that the journal Phytokeys listed those 2 species as 2 of their top 5 papers that they've ever published. They've not been around for that long, but that was pretty neat, you know, the sort of relationship with a journal and a journal that has a media office, this sort of ability to work with someone there and promote papers has been really beneficial for us in this case. So I'm pretty psyched about it. And of course, you know, we've continued to find some new species and work with them. Not all of them have been as successful as sort of case studies in media. We haven't really pushed them like we did the other 2, but this is one we named after another botanist named Pete Jobson, Solanum jobsonii. This ended up with one really amazing outcome, which is that there was a yarn named after him, basically. So the Ixchel Angora Fiber Farm named a new yarn color, the Sexy Botanist. And when you go online to buy it, it describes how this species was named. Here's one of the photo plates from the paper. How the species was named for this guy, Jobson, who actually is a really well-known knitter in Australia among the knitting community. And so here's the Solanum color palette, right? It's the green plant with the corolla and the anther color. So that was a really fun outcome as well. And we have a new species that we are that's out for review right now. And this is one we're calling Solanum plastisexum. Lead author is Angela, who you see there. And we have a great undergrad named Heather Whitdrive who's on this paper as well. And this is one where we're trying something a little bit different. This is a species that over the years has been really difficult to place because of its plasticity in terms of the way its sexual form shows up. So sometimes you run into individuals that are male, sometimes they're clearly hermaphrodites, sometimes there's something called andromonaceous. So these three different sort of sex forms that turn up, and it's what's led to it being difficult to describe and give a name to. We see this as a really great model for the fact that plants do all sorts of things in terms of their sexual behaviors. That's true for plants. It's actually true for most organisms on the planet. So we are using this. We have a press release that's going to come out with this paper that talks about the fact that sort of classical notions of some sort of binary sexual condition are totally a fallacy, and that plants are a great example of that. So we're taking a bit of a different angle on this one, but I think it's going to be really cool to get that message out. And more things we've been working on. Things from Hawaii, including a new subspecies of the spinach relative, one of those, that Chenopodium from Molokai. Yep, and some new nightshades and some other things. And you might get the notion, and I'm talking to a very educated audience, but people might get the notion that the way to find new things is to go far away in exotic places, right? And we've done so with these Australian things, at least. But my last story I'll tell you is actually something that is more regional, and for me, very local, right? And this is where I'm going to go back to that most recent episode of Plants Are Cool Too that we've made. The focus of this was a really unusual cliffside habitat that's just 10 minutes from the campus where I teach. And another focus of this was to sort of look at the sort of close relationship that we can have between non-academic and academic botanists. So I work very closely with a guy named Scott Schutte with the Pennsylvania Natural Heritage Group, and we are doing a lot of work together now, looking at conservation of rare plants in Pennsylvania. And one of the sites we decided to go look at was this cliffside habitat. We went there initially because there's a corydalus that lives on this cliffside and nowhere else in Pennsylvania. So there's a single site for this one corydalus in Pennsylvania. It's this cliff. So we went there to sort of see if we could find it again. We went there to sort of show people how heritage botanists do surveys, how they assess how a rare plant is doing. We got a bunch of ropes from our climbing wall folks at Bucknell and got on those ropes and went down. Luckily, Jay Cantley, who's now at San Francisco State, but is my former postdoc, did some cliff work in Hawaii, so he knew what he was doing. And we went out there and looked around. And you can see sort of, there's Jay hanging on a rope here. So these are about 350-foot cliffs. Sorry, maybe 100. I said that wrong. There's Scott climbing up. A bunch of our students were out there as well. And over the course of the day, I was just taking pictures and putting them out on Twitter and saying, hey, we're filming this new episode, just trying to generate some buzz about that. And I put up this photo that I thought was Heuchera americana. It had a bumblebee on it. I was pretty psyched about that. And I put that out there in the world and said, yeah, here's this species. And pretty soon after that, Ryan Folk, who is a global expert in Heuchera, who's at University of Florida, responded and said, that's not americana, that's something called Heuchera alba. And then, showing real enthusiasm for a botanist, used 2 exclamation points on quite rare. And I thought, oh, this is a big deal if he's doing that, right? And then I said, God, this can't be. It's not known in Pennsylvania, right? And then I sort of bring in Scott Schutte, whose Twitter handle is the best. It's @Mossman2000. And I say, Scott, you better check this out. You might be interested in this. And Scott says, yeah, check the specimens. This is really important to conservation. I knew he was excited because he said #rare species, right? So I knew we had something really interesting here. So what did I do? I went and checked the specimens in the Bucknell Herbarium. We have a small 20,000 specimen collection. And I went in and I looked at our Heuchera collection and for things that were collected in that region. And what I found was that we actually did have some specimens that had been misidentified from around that place. And one of those had a label on it that had been collected in 1905. Very little information, but the only locality information it had on it said Gundy's Farm. And I thought, why do I know that name, Gundy's Farm? And I remembered that one of the few things left in my desk when I took the job at Bucknell 7 years ago was this photo showing a field botany class. And on the back of it, it said Gundy's Farm. This is from 1912. It said Gundy's Farm. This is how we still do field botany at Bucknell, FYI. I require the same dress code. If you can't see it from there, they're wearing suits and bonnets and dresses and things like that. Right? And so I thought, oh my gosh, I actually know where that spot is. I know that railroad track. I know that cliff. Let's go check it out. So Angela McDonald and I walked 10 minutes from campus down the railroad tracks up to that cliff. And we looked up and there it was. Euchera alba in the same spot where it had been collected in 1905. So people have been seeing this thing and collecting it for over a century and just calling it the wrong thing. And then we went out and looked at other parts of Pennsylvania that had the same geology, same pH, same sort of exposure. Scott took the sort of western part of PA. I took this Susquehanna River Valley, and we found it everywhere you see a yellow cross here are new localities for this species, Euchera alba. So this is the first time we've ever found this thing in Pennsylvania. Before we filmed and I got it wrong, it was only known from a few localities in the mountains of West Virginia and Virginia. It was already considered globally imperiled. I would say that our 6 or 7 new populations don't really take it out of that category. They're not very large. But here you have this really cool range extension of an already globally rare plant that was found because I posted it on Twitter and called it the wrong thing, and a scientist who knew better actually saw it and had a comment. I'm very excited. We were super excited about that. I can tell lots of people about this that are not scientists because everybody has coral bells in their garden. So this is also a cool thing for me to be able to tell this story, get people excited about this sort of stuff. And we were so excited about the range expansion and other things about it that we wrote a paper and had that published about this. I was super psyched that they let me put Twitter in the title of the paper. And that became a fun story, that we had discovered this rare plant on the cliffs in Pennsylvania because of Twitter. So we made the front page of the newspaper. The local news did a story on it. It opened the 7 o'clock news. It was pretty awesome, like live from Bucknell University, a rare plant found on the cliffs. It was a really good, nice, positive way to start the news broadcast. And that got us in places like IFL Science and Earth.com and on the Quasi Ecology Community blog page, and even a mention in the dispatch section of Frontiers in Ecology and the Environment, which was really awesome. My favorite was this story from Quartz.com, if only for the subheadline, Twitter, sometimes good. Right? If there's anything I want you to take from this talk, is that Twitter and the use of social media can sometimes be good, right, for biodiversity science. The best placement for this by far was that we ended up on the Science in Action podcast for the BBC World Service, and I knew this story was big because it was placed in between a Japanese space agency mission and koala poop transplants. And there we are, right in the middle of those 2 big stories, right? So I knew we had hit the big time. But that was actually really nice. It's a really nice piece about the sort of scientists using new forms of networking to share information and make discoveries as a community. Something I'm really excited about. So yeah, did it turn people to the paper? Yep, 2,900 views on a paper that maybe 5 people would have read if we hadn't done something like this. So I'm really excited about people turning to the primary literature and at least seeing what a scientific paper looks like. They're not scientists already. And it generated some cool buzz. We ended up being featured, the species ended up being featured in a couple of different ways. The PA Botany Symposium last year had it on its t-shirt because we were excited about it. Somebody did a painting that ended up being part of the promotion for a local land conservancy that actually had recently transferred a bunch of the cliff face to a local state park. So that actually led to conserving some of the habitat that this rare species is on. They didn't know it at the time, but now we do. So they commissioned someone to do artwork. And because of that, the artwork appeared on a banner hanging in the middle of our town. And I did scream and pull over and jump out and take that photo because it's just such an exciting thing to have people, that were enthusiastic about this cool new find. So I do think that people really actually do care about this stuff. I think people want to learn about these things. They will care, but we have to let them know about it, right? And as much as it's great that there are these opportunities to communicate science to a broad global audience in a way that is unprecedented in our history, it's also true that access to information comes with a lot of garbage. And so there's all sorts of junk that's in our faces all the time. And I think as scientists, I think to some degree, it really benefits us to put some good stuff in there as well, right? And if we can put good biodiversity stories in front of people's eyes, because they're in those electronic spaces anyway, there are ways that we can still get people excited about it, whether it's a t-shirt or yarn or some artwork, and maybe even drive into some of the primary literature and maybe inspire some young people to actually turn to science as a career choice. Right? So I think there's a lot that can be done. That's why I wrote this piece that I'm happy to share with anybody, because I do think it's okay for us to do this. Right? I think it's actually okay. It's high time that we embrace this as something that can benefit us and as individuals, in ways that you might imagine, but also as a community of scientists that are doing really good work that people need to hear about. So embrace new outlets. Not all of these is necessarily new, but get out there if you're willing, right? And don't sort of be offended if you're not into it and other people are, because I do think it's actually a wise use of some of the time. I want to end with this last thing, and that is that a lot of the work that I do may be seen as encouraging people to spend more time looking at screens. And I want to just acknowledge the fact that the really, truly transformative experiences that young people can have are their outdoor experiences in real places with real organisms. I will also, of course, include museums in that, right? Seeing real things and learning about real things is an unparalleled way to get young people excited about biodiversity. I see that with students I take out in the field for courses, for research. There's a lot of smiling faces there, and that's because once people's eyes get open, once we begin to sort of will away at what we've all been calling plant blindness, the whole new world opens up for people, and we can still inspire people in ways that we're all kind of programmed to be inspired. So I think there's a lot of hope, and I think if we just do a little bit of legwork, we can get people excited about this stuff. So thank you for listening to me. I'm really glad.
Open to questions. Any questions?
Yeah, so scientific papers have a lot of jargon and a certain style that we're comfortable with that we expect, but that non-scientists find really off-putting.
Yeah.
Has this affected how you write papers? Like knowing that people might actually be reading your paper that are not scientists?
Yeah. I'd say to some degree, yes. It's not coincidental that the papers that we've really pushed in this way are new species descriptions and things like that floristic treatment, because they are a little bit more accessible. And what I have found is that what I tend to do, especially with new species descriptions, is have a discussion section that really is just a couple of readable paragraphs, right, for anybody that reads it as a summary of like, why is it important that we found this, how is it different, what's interesting about it, right? So yeah, to some degree I have, knowing that there's going to be sections of every paper that a layperson's not going to want, right? But it's a sort of that same point. The papers that we're writing that are sort of phylogenetic papers, the big, dense papers full of trees, are not the ones that I'm choosing to promote. Right? I still want people to read them. They're going to end up being cited more, probably, right? But they're not the ones I'm choosing to sort of push in sort of a public. Right? I'm also a person who's been dinged a lot of times in my career for writing casually in some scientific papers. I've been trying it for a while.
Two questions. How important is it to have good visuals or graphics? And then the second question is, how important is it for the article to be open access if you're going to be pushing it through all these?
Yeah, great questions. The Ossi cruentum, the sort of spiny, bloody thing, that story was really made by the images. Jason Cantley, my postdoc at the time, took some really great photos, and those went out with the press release, and they made a huge difference, right? Having those images. So it's important to think about that too. When people are just seeing stuff come across, it's not going to be the text that gets their attention. It's going to be some sort of thing. So I think it's really important. It's why I'm always encouraging students to constantly take photos of stuff as they're doing the research. So you have this stockpile of stuff, not just for the paper, but for other things, like talks and other things. The open access thing is interesting, right? I think in our case, it's really benefited us, but I also have money to pay for page charges, right? It's expensive to sometimes to publish open access. Obviously, there's a push to try to make people go open access right now, and I think it's not possible for all journals to offer that all the time, right? A lot of society-run journals, some of my favorite journals, it's hard to do. So having the open access thing is, for this kind of stuff, is actually pretty important, because otherwise people are not clicking and going and finding the paper. It would be behind a paywall. So I'm torn. I feel like lucky that I can actually pay for my stuff. The open access when appropriate, but I also understand that it's a thing that not everybody can afford. I'd like to see some sort of model where we're still able to support some of our traditional journals and also get things out of that. I think we're approaching that, but so clearly I've been taking advantage of it. Yeah.
I
I have a question about, just a concern about the heuchera that is rare.
Yep.
And then doing open access, and now everyone knows kind of where it is. And there's probably not very many people that are gonna be protecting or monitoring that, or are there, or how
Yeah.
what are your concerns about that?
Yeah,
Can you repeat the question too? for
Sure, yeah, the question was about sort of publishing open access and publishing locality data for this rare Heuchera alba. The sites that we were working in that we sort of spent a lot of time talking about are on state parkland. And one of the benefits to the habitat is that it's on these very steep
Right.
cliffs, right? So the reason we got to them is that we were on ropes, so we were rappelling off of a steep cliff to get to them. So we did sort of talk to people in state parks, we did talk to heritage people where, Scott is in the heritage program. We did talk to the conservancy that sort of is working on some of those lands about this, whether we should do it at all. They all felt pretty good about doing it. It's hard because our sense is that this species is actually more abundant than we're finding. It's just difficult to identify. So we wanted to get the information out there so that people can go look and find more of it. And we think it's probably way more abundant than we currently know. It's a real bugger when it comes to ID. So we have a key in there we're hoping people will use, and some illustrations and images. But we did think a lot about that.
How do you, the scientists who are in
We have in plants are cool too. How do we, maybe it's more like, how do we pick episodes? How do we get scientists? How do they get us too, right? And so I've been going to the Botany conference every year for, feels like 100 years. And since we started this about a decade ago, talking about it. And initially it was sort of, me just saying, okay, what are some interesting stories and interesting research questions that people have in cool places, and where some of the people, at least involved in the science, are also likely to be pretty good on camera. So sort of trying to find these sort of sweet spots where there's a story that would be easily translatable to a broad audience with good visuals, so in a cool place or some other thing, with people who I think, would be good in the interview. But my job, this is something that I was talking about with Peggy earlier, my job as being host is that I'm an informed host who can get that stuff out of people, right? Be a bridge to the audience. So it's something I take pretty seriously. I think just about anybody can handle it, even Warren.
Yeah.
That's why we're doing it next week, because he's not great talking to folks.
No. I'm trying to say something.
Go ahead.
Another aspect of that is, for like this set of Hawaii ones, is some grant agencies, like in this case, National Science Foundation, wants outreach component to your research. So we developed a suite of things that involves students, conference that we're trying to get conservation people in Hawaii to attend. And then for the third leg of that stool is doing a couple of plants or two videos, so that we have three different kinds of approaches. And NSF apparently loved it because they gave us a whole bucket full of money.
Yeah, so that's another model that we're trying to embrace a little bit, is to get people to include us in their outreach plans.
And as it
some of their
becomes more
external
popular,
plans.
more people will.
Yeah, they may, right? So there's a couple that have written us in the first edit this month. It was yours, so yeah, that's another model. And one of the things that's tough to balance is how much of my time am I going to have as a researcher and an instructor, professor, and then also maybe teachers, right? So I've sort of been like, one or two a year if we can find the funding. But I'm always up for ideas, right? I can't know everything in school that's out there. So to some degree, it's all those things I mentioned, plus a balance, sort of like capturing the diversity of people who are doing botany work, another thing I think a lot about. So there's a lot of things, but got ideas. Jen, you have?
I have a science question. So for the Spanish, the bleeding tomato.
Yeah.
Is it related to the South American spiny tomatoes?
Yeah, so this is part of that big subgenus called Leptostomonum, the spiny solanum. They're really eggplant relatives. That's
Is
probably
it
the closest
only
cultivated
in
thing.
Australia and South America?
No, mostly Southern Hemisphere, but pretty much everywhere. But most of its diversity is South America, Africa, and Australia. Not a lot of stuff around the rim of the Indian Ocean. There's not very many access to those, but some Asian distribution, but not a lot.
Because we just found a small group of Asian grape species. The closest to the relative is in the southern part of South America.
That's pretty cool. Yeah, that's pretty neat. Somebody asked me actually yesterday about new, sort of, where did you say the grapes are from? Did you say New Guinea?
It's in New Guinea
Yeah.
and one species in Australia.
Yeah, somebody was asking me about whether New Guinea species are closely allied with stuff in Northern Australia. For us, it's not. It's a very different sort of lineage. Any other questions? Cool. Well, thank you
Okay.
again for having me. This has been a ton of fun. So thanks, everybody.
Thank you.
Oh, next slide. So there's the information that you can call into the PCA meetings. That I should be saying. There's
Yeah.
the number. You can see it there.
All right, great. Thanks a lot, Chris.